In this video, we're going to be looking at some of the ways that we move materials across a cell membrane. And we'll start out by talking about passive processes. Processes that don't require any kind of cellular energy in order to move a material from one side of a membrane to the next. So let's get started on this. So the first thing we need to talk about is membrane permeability, because the cell membrane, the outer boundary of a cell, is what we call selectively permeable. If something is permeable, then that means that materials can pass through it. So we could think about like a screened window is permeable to the air. The air can move through the window screen, but it is impermeable to mosquitoes, for instance. So if a membrane is selectively permeable, that means to some materials the membrane is freely permeable. Those things can pass through the membrane with no problem at all. So that would be like the air. A window screen is freely permeable to air, wind passing through it. With a cell membrane, oxygen, so O2 gas, carbon dioxide gas, and water, as well as anything that's a small lipid-soluble chemical, is able to pass through the phospholipid bilayer. And that's because, if we were to draw the bilayer here, oxygen and carbon dioxide can dissolve in lipid, and so they are able to just slide right on through. Lipid-soluble materials, that means that they dissolve in lipid. They can slide through as well. And water can pass because there are these cool little channels called aquaporins, and they're made up of proteins, transmembrane proteins, that allow water molecules just to slide right through the membrane. So it's freely permeable, this membrane is, to water, oxygen, carbon dioxide, and other lipid-soluble molecules. But to most materials, cell membranes are impermeable. That means that those things cannot pass through the membrane. And so some examples of what a membrane is impermeable to include any ion, any charged particles. So magnesium 2+, chloride ion, sodium ion, they can't pass through. So if it has an electrical charge, it doesn't pass. If it's really large, it doesn't pass. If it can't be dissolved in lipids, it won't pass. If it has a shape that is incompatible with being able to pass through the lipids, then it won't be able to pass. So the permeability of a material is all determined by these kind of factors or a combination of those factors. All right, so cell membranes, semi-permeable, because some materials get right through it, other materials cannot. And there are two general ways that materials are able to make it across a cell membrane. Through a passive process, also thought of as a physical process, and through active processes, also thought of as physiological processes. So passive processes, that means that it's the movement of materials across the cell membrane without expending any kind of energy. So no ATP is required. And materials move across a cell membrane passively because of the physical properties of the membrane, the molecules that are moving, and the environment. So it's just a physics thing. It has nothing to do with anything biological. It's a non-living process, and it's all because of the inherent, what we talk about as kinetic energy of the materials that are being moved, and the concentration gradients. So if it's more concentrated on one side of the membrane than the other, and pressure gradients. So if there's high pressure on one side of the membrane and low pressure on the other side, then that will cause movement. So all of these are non-living components. There are non-living characteristics that cause the movement of materials across the cell membrane. And so there are a number of kinds of movements of material that are this kind of passive process. Diffusion, osmosis, a process called facilitated diffusion, and filtration all work to move materials from one side of the membrane to the other without expending energy. Now these active processes, they all require ATP, and they are living processes that can only be done with living cells because they employ changes in the cells and changes into the proteins that are in the cell membrane. And they include things like active transport, endocytosis, exocytosis, and then another process called transcytosis. So we're going to start by looking at these passive processes, the physical processes that move materials from one side of the membrane to the other. So first let's talk about the idea of simple diffusion. So this is exactly what we were doing with the dye molecules, with the food coloring molecules in lab recently. So if we were to drop some dye molecules, some food coloring into water, over time, because of the random movement, the kinetic energy that's associated with any material that's in solution, all the molecules are randomly moving it about, and they collide, and they bounce off of each other, and they go in opposite directions. It's almost like two pool balls. So if we have a pool ball moving this way and another pool ball moving this way, when they meet and crash together, they will then move away from each other, going in opposite directions. So the collisions that allow this to happen, water is a pretty small molecule, but dye molecules tend to be much larger. And so those collisions, when two dye molecules collide, there's a whole lot of movement that happens. If we were to slam a ping pong ball against a pool ball, we would get very little movement of either of the ping pong ball. A lot of movement of the ping pong ball, but the pool ball wouldn't move very much. So anywhere that there's enough concentration of dye so that the dye molecules are going to be slamming into each other, we're going to see movement away from that area of high concentration and movement toward areas of low concentration of the dye molecules. So over time, because of all these random collisions that are happening among the dye molecules, we'll see that the food coloring molecules are going to wind up being evenly distributed throughout the solution. So with simple diffusion, we move molecules from regions of high molecule concentration to regions of lower molecule concentration. So the dye moves from where it is most concentrated in that drop that falls into the water and spreads out evenly throughout the jar. Just not because anybody's stirring it up, not because anybody is mixing it or doing something to move it around. It just does it because of the physical properties of these wiggling molecules that will collide into each other and bounce away, headed in opposite directions. Simple diffusion. Now, if we were to put a permeable membrane to separate the beaker into two compartments, what would happen? So we put on one side a whole bunch of the solute. It could be dye molecules. You could think of it as sodium and chloride ions. All those molecules that are in there mixed in with the water. And we have a permeable membrane that allows the solute, if we have a solution, there are two components to a solution. A solute, that's the material dissolved in the solution. SOLN with a little apostrophe there. That's my abbreviation for, come on, that's my abbreviation for solution. Well, that's an apostrophe, believe me. And then there's the solvent. Come on. That's the material dissolving the solute. So we've got a solution here, and it's got a lot of dye molecules or a lot of solute on A side, and there's no solute on the B side. So what's going to happen if the solute and the water molecules are able to pass through that membrane? Both of them can pass through the membrane. I want you to think for a minute what you would expect to have happen based on simple diffusion. So you can pause the video, spend a little time thinking, and then start the video back up again, and we'll talk. Okay, so I'm assuming you are now ready to see the answer. So here it is. Over time, the two compartments in this beaker that are separated by this permeable membrane, they will reach an equilibrium where both sides of the solution, both compartments of the solution, are going to have approximately equal concentrations of both the solute and the solvent. Because water that's in high concentration here on this side is going to move toward this side where it's in lower concentration. The solute, where it's in high concentration here, is going to move to this side. Dadgummit, I'm having a hard time here. There we go. The solute is in high concentration there on this side, and it would move to B because of moving from areas of high concentration to low concentration. So over time, with all this movement back and forth, we're going to see that the two compartments will come to an equilibrium in which they have similar concentrations of solutes. And it's simple diffusion that moves carbon dioxide out of red blood cells here in the lungs. Oh, I'm sorry. It's yeah, I think I've got a bad description here. I'm sorry, I got confused. What we are seeing here is a cell out in the tissue. So here's the cell where the mitochondria are busy making ATP. And so here in the cell, there's a high concentration of CO2. As the blood out in the peripheral tissues goes past that cells, the CO2 is going to move from the cell and into the blood. Because it's being produced here in the cell, it's going to have the highest concentration in the cell. But the blood that's coming past, it's blood that's coming from the lungs. It was just all the CO2, or a lot of the CO2 in that blood, was breathed out at the lungs. And now this low CO2 blood comes by the cells. And the CO2 moves by passive diffusion from a region of high concentration to a region of low concentration. Now the blood that's coming from the lungs is also going to be really high in oxygen because that's the source, the lungs are the source of oxygen in the body. And so that's high O2 blood coming past the cells, and passive diffusion will move it into the cells because the oxygen is being used up there. And so if we're using up the oxygen, then it's always going to be in the lowest concentration there inside the cell. And this is all done with no energy being expended to move the oxygen from the blood into the cell or to move the carbon dioxide from the cell into the blood. It's all because of these physical properties that relate to simple diffusion and the movement of materials from a high region of high concentration to a region of low concentration. Okay, simple diffusion, nothing is required to help move it, but there is also a process called passive diffusion. By the way, if you hear a little jingling in the background, that's my cat. He's got his collar on and he's gotten real active all of a sudden. So he's bouncing around and that's that jingly noise. So sorry about that if it's distracting. So there's also a process of diffusion that can move materials from one side of a cell membrane to another. But in order to get it from one side to the next, it has to use something called a carrier molecule. So here we've got some kind of material that cannot pass through the cell membrane. It would like to get through, but it's blocked because of the oily nature there of that cell membrane. But it turns out that embedded in the cell membrane are transmembrane proteins that are capable of binding this solute here that's on the outside of the cell. And when it goes and binds in here, that causes the molecule, this carrier molecule, to change shape. So where it used to be open on the outside, the shape changes and it closes on the outside, opens on the inside, and releases the molecule, or the material, into the cell. So it's this awesome process where a protein carrier molecule, that's what these purple things are, will change its conformation, and in the process will move a solute from one side of the cell membrane to the other. No ATP is required for this to happen. It's really cool that we've got these molecules that, if they can bind a specific molecule or a specific material that they interact with, if they bind it, it causes their shape to change, and it dumps that material inside the cell. How cool is that? And it will remove materials from regions of high concentration to regions of low concentration. So it's facilitated because it's helped out or facilitated by the carrier molecule, but it's still diffusion because no ATP is being required, and we're moving from a region of high concentration to a region of low concentration. As soon as we drop off whatever the material is that was moved, then the carrier molecule snaps back into the first conformation, where it's open, waiting, hoping to be able to bind this molecule, whatever it is, out here. Okay, so that's facilitated diffusion, and it moves things from regions of high concentration to regions of low concentration. But in order to do that, it requires a carrier molecule. All right, so we've talked about simple diffusion and facilitated diffusion. In the next lecture, though, then we will talk about a special kind of diffusion that moves water from one side of the cell membrane to the other. So that's the end for this one, for this lecture. But in the next one, we're going to look at the last kind of simple diffusion called osmosis.